Science Uncut, presented by the Volkswagen Foundation.
How healthy we are depends on what we eat. This is something we all know. The connection between our diet and our health, however, becomes vastly more interesting and complicated when we ask how we can prevent or cure diseases by changing what we eat. Food allergies, type 1 diabetes, autoimmune disorders, asthma, cancer, or cardiovascular diseases are all influenced by our diet, says Charles Mackay. He holds the chair for diabetes at the Charles Perkins Centre at the University of Sydney in Australia. In his talk, he focuses on the molecular mechanisms by which fibre in our diet protects us from diseases. What we eat, says Mackay, has a profound effect on the gut microbiota. To find the origins of many of our so-called Western lifestyle diseases, we have to look not only at what we ourselves eat. It seems that the composition of the bacteria living in our intestine depends on the bacteria that cross the placenta in utero. Our health depends on what we eat and, research suggests, also on what our mothers ate when they were pregnant. Diet, gut microbiota, and Western lifestyle diseases is the title of Charles Mackay's talk, which he gave on October 9, 2014, at the Herrenhausen conference Beyond the Intestinal Microbiome, From Signatures to Therapy. The conference was organized by the Volkswagen Foundation and took place in Hanover, Germany.
Thank you, and it's an honor to be here. I'll start with the first slide, and I think we're pretty much all on the same page here. The main point I want to make is that things like diet, particularly high fiber or high fat, are having a profound effect on the composition of the gut microbiota, and this in turn can have a bearing on human diseases. And we personally believe that many of these Western lifestyle diseases, such as food allergies and type 1 diabetes, asthma, IBD, and perhaps many others, cancers, cardiovascular diseases, and so forth, are relating to this connection between diet, gut microbiota, and factors produced there that somehow influence these diseases. So I think moving forward in this field, it's critically important to understand the cellular and molecular pathways that are mediating or possibly mediating these conditions. And that's going to be the subject of my talk. So I'll also begin with this other cartoon, which really concentrates on dietary fiber, because we think that a lack of fiber is one of the critical things in Western diets that is causing dysbiosis and eventually pathways that lead to some of these conditions I just mentioned. In fact, one of the main roles of commensal bacteria is the digestion of fiber, because they have the enzymes to do this, whereas we don't. And one of the products of the digestion of fiber is short-chain fatty acids. And I'd like to stress that these are produced in enormous quantities in the GI tract, particularly the lower colon. You know, for instance, acetate is at around 40 millimolar. And I've outlined in this slide many of the actions of short-chain fatty acids in the gut, all to do with gut homeostasis. For instance, the stimulation of immunoglobulin A production, which we've already heard about today, stimulation of mucus production, tissue repair, which we haven't published on yet, but a beautiful effect of high-fiber diets in tissue repair and wound healing. What I'm going to concentrate more on today is some of these things at the bottom of the cartoon. For instance, epithelial integrity associated with inflammasome activation, and also I think a very exciting development in this field, which is promotion of Tregs and the possibility of inducing immunological tolerance to antigens of choice. So we have really seen quite remarkable results when we play with diets. If we look at a really high fiber diet, which we manufacture for use in mice, or if we take fiber out of the diet, we see, virtually in every model we do, quite incredible effects. Here is in a DSS model of colitis, If you just look at the histology first, this is the model in a fibre completely, in a mouse completely deficient of fibre, and you can see the destruction of the mucosa compared to a mouse which we fed a very high fibre diet to. So we see this very impressive gut homeostasis effect with high fibre, and you can also see it just by looking at the colon lengths in the zero fibre versus the high fibre mice. So I wanted to sort of just cover some of the molecular mechanisms whereby fibre could be protective, and I should also stress that fibre is protecting in various models that we look at, from asthma to food allergy to cancer models, type 1 diabetes. It's just really quite remarkable. And I've listed the three main molecular mechanisms where these bacterial metabolites or just dietary metabolites impact on human health or mouse health. And that is particularly this growing class of receptors, the metabolite-sensing receptors, which I'll cover in a minute, histone deacetylase and their inhibition, and also transcription factors like the aryl hydrocarbon receptor. So the way we fell into this field was we started looking at a mouse that came out of, a gene that came out of transcript profiling called GPR43. And I think this is an archetype-type metabolite-sensing receptor because all of the other ones look very much similar to this. And when we looked at a knockout mouse for GPR43, we got this horrible, exacerbated, non-resolving colitis in the DSS model, as well as other colitis models, compared to sort of the perhaps more normal inflammation and destruction you saw in a wild-type mouse on DSS. And I should also add, we supported this result when we went to, rather than the receptor knockout, we went to a ligand knockout when we used germ-free mice, which are devoid mostly of short-chain fatty acids, and we got the same result. I should mention that 43 is one of the main receptors for short-chain fatty acids, the products of the digestion of fibre. So in fact, 43 is just one of many metabolite-sensing receptors, and in fact, all of these receptors show a remarkable similarity. There's a number that bind the short-chain fatty acids, like 109, 41, and 43. Then there's receptors, these sensing receptors, for most of the sort of healthy foodstuffs that I would call them. For instance, omega-3 fatty acids binds to GPR120. Then there's tryptophan metabolites, such as kynurenic acid, which binds 35. And when we start looking at a number of these receptors, you get like the similar result. You get exacerbated inflammation, particularly in models of colitis. And also you get metabolic phenotypes. There's been many beautiful papers on the knockouts of a number of these receptors where metabolism is dysregulated, and often the mouse will get obesity. So it's very likely that any mechanism going through the microbiota or through a dietary metabolite will involve one of these classes of receptor. GPR109, a similar receptor to 43, perhaps even a stronger phenotype in DSS colitis. And I just wanted to emphasize in this slide that when we feed high fiber to these knockout mice, we cannot improve colitis, as shown nicely by histology. So there's no beneficial effect of fiber in the 109 mice or in the 43 mice, which we don't fully understand the redundancy there. So these two receptors are mediating the protective effect of high fiber at least in the DSS colitis model. So we sought to understand how this was happening. And this is a cartoon that summarizes 20 or 30 minutes worth of data slides. So I'll just walk you through that. But basically what we were able to do is show that signaling through these metabolite sensing receptors connected through to the well-known inflammasome pathway, which has been well characterized over the last 5 or so years as one of the major pathways for gut homeostasis. And in fact, what is happening is that short-chain fatty acids, which are produced by the digestion of fibre, for instance, acetate, butyrate, and propionate, are binding these metabolite-sensing receptors such as 43 and 109. For the inflammasome pathway to work, you need two signals. You need, we're not quite sure what the signal is for epithelium, but presumably some sort of fecal product or a TLR ligand or something, which we can produce by just stimulating cells with fecal material. So there's a signal one, which is particularly strong in healthy microbiota, and then you get the signal two through the G protein coupled receptor signaling, which characteristically produce G protein signaling calcium efflux, which is known to activate the inflammasome pathway, particularly NALP3, which we find then is sufficient to lead to the cleavage of pro-IL-18, which is an inflammasome-related cytokine relevant for epithelial integrity. But and the way that these receptors, these metabolite-sensing receptors, do this is, I think, through conventional G protein-coupled receptor signaling. But I should also emphasize that all of the metabolite sensors signal through another pathway. We're not quite sure the circumstances between the different signaling pathways, but the beta-arrestin 2 pathway is well characterized for its inhibition of NF-kappa B. And pretty much every single receptor that's been studied that I've mentioned in these previous slides shows this characteristic beta-arrestin 2 signaling. I'd like to also stress that these metabolite-sensing receptors, which I think are incredibly important, aren't the only mechanism where metabolites will influence impact on biology. For sure, this is an immediate response because it'll happen, in seconds or minutes. But then there's also the transcriptional and epigenetic effects that many of them, well, certainly the short-chain fatty acids can have on gene expression. And short-chain fatty acids are well known histone deacetylase inhibitors, which impacts on histone structure and transcription. And inhibition of a histone deacetylase inhibits the deacetylation of acetate, which profoundly affects gene transcription. And it also affects the acetylation of individual transcription factors, which will impact on certain gene expression programs, which I'll come to in a minute with FOXP3. So this latter pathway, I think, could be particularly important for epigenetic imprinting. This slide shows some of the points around the body where bacterial and dietary metabolites impact on the immune system. And of course, the most obvious one is in the gut. But one I think that we haven't really talked about in this meeting is this impact on fetal development and the fetal immune system and even other tissues. Because many of the bacterial metabolites will be produced in the colon, enter the blood, and then cross the placenta into the fetus. So, in fact, I don't know how many people here are aware that, in fact, this is a really exciting and I think relatively new field. Well, I wasn't even aware of it until a year or two ago, and that is this whole developmental origin of disease. And many people think that many of these sort of Western lifestyle diseases, particularly cardiovascular disease, but possibly also obesity and diabetes, and I would argue asthma and others, in fact, have their origin in utero and early life. And we got data to support this concept when we, I think, got quite an amazing result. What we did was we did an asthma model with house dust mite, and we fed pregnant mice very high levels of fibre or acetate in the drinking water, and then we stopped this before birth, And we made sure that they we did a placental transfer to make sure that they just weren't inheriting a certain microbiota. And then we just let the mice grow up. And so these mice have never seen a high fiber diet or the microbiota from that. And then we did the asthma model. And we could, like for instance, with acetate in the drinking water in the pregnant mice, we could not induce the asthma model, the typical response exemplified by eosinophil numbers in the airways, and also markedly reduced IL-5 and IL-13. And also, incredibly, just looking at the airway responses using metacholine challenge, we could not get a response in the airways in response to house dust mite at all in these offspring that had never, the only thing they'd ever seen was metabolites, presumably from their mother, that had crossed the placenta during her pregnancy. So I'm not going to show you all the data that came from this, but so first I'll show you that we've got also data in the humans to support this. When we've looked at short-chain fatty acid levels in human mothers, we've also correlated that to the amount of dietary fiber they were eating. So the more dietary fiber you eat, the higher the concentrations of short-chain fatty acids. This is actually not a particularly good representation, but in the literature there's actually much better ones. And in fact, the mothers that were eating very high levels of fiber or were showing high levels of short-chain fatty acids had children that had these symptoms that is suggestive of a low incidence of asthma. This is somewhat preliminary, but we're very keen to follow this up to see if this spectrum of high acetate versus low acetate in developing fetuses and in the cord blood then is a predictive of going on to develop asthma later in life. So I won't show you all the data, but in fact, this is our model. Acetate in particular, which is the main short-chain fatty acid that crosses into the blood, can also cross into the fetus. We showed that. And it has these effects on histone modifications, particularly the inhibition of histone deacetylases. We've shown that it's in particular HDAC9. And this is an important histone deacetylase for the regulation of FOXP3. And in fact, so we show in this study, which I'm just summarizing here, that this is particularly important for Treg numbers and Treg activity, which is one of the main pathways considered important for the development of asthma. But one of the funny things about asthma, I mean, the field's kind of split. People think it's an immune regulation disease, and there's a lot of other people that think that it's, in fact, an airway epithelial condition. And in fact, FOXP3 is expressed both by T regulatory cells as well as by epithelium, including in the lung. And a target gene of FOXP3 that we showed could be modified by our acetate in the mother was this gene that in fact had been implicated in the pathogenesis of asthma. So we think that we're setting something up in utero, changing gene expression and lung function, as well as T regulatory cell numbers and function at this early stage, which can then have a bearing on asthma later in life. So I've sort of given you a flavor of the short-chain fatty acids, the metabolite-sensors, a little bit on the epigenetic effects that could be mediated by metabolites. I just wanted to briefly mention this pathway because I think it's so important, and there's so many studies that have emphasized its relevance. So in fact, tryptophan metabolites that come from foods such as broccoli and eggs and fish and meat are digested, and these typical pathways, in this typical pathway to things like kynurenine, kynurenic acid, and also niacin. And it turns out that many of these metabolites have either sensors, G protein-coupled receptor sensors, or transcription factor sensors for all of the members of this pathway. And there's also an important transporter for tryptophan. and also the gut microbiota can be involved in the digestion of tryptophan to an AHR agonist. And there was a number of beautiful papers sort of all showing the relevance of all of these factors for gut homeostasis, including this nice paper from Joseph Penninger in Nature on mice deficient in this tryptophan transporter, and also this very well-cited paper on the aryl hydrocarbon receptor knockout, which basically gives a DSS colitis picture similar to what I showed you for some of the other metabolite sensors. Now, we have studied, and I've already shown you GPR 109, which is also exacerbated colitis in the knockout. So this is, the presence of this receptor is protective. We've also got similar data for GPR 35, which we haven't published. Interestingly, 35 is one of the stronger genes that have come out of GWAS for human IBD. So to move forward onto sort of therapy, and this is, I suppose, a strong theme of this session, this conference, we have, I suppose, a lot of enthusiasm for the possibility of either preventing disease through modulation of immunological tolerance, for even treating autoimmune diseases, and other sort of autoreactive diseases such as food allergies. So I'm just going to quickly run through some of the things that we've done here. We had a hint that some of these metabolite sensors were involved in Treg biology just by transcript profiling. We got nice expression of some of the receptors like 65 and GPR sort of 41, 109, to a little bit less 43. And this is just on Tregs, and not to mention cells upstream of Tregs, such as tolerogenic DCs and so forth. And then we saw that the 43 knockout mice were deficient in Tregs. And then these three papers came out, this one from Smith in Science, showing that short-chain fatty acids, such as propionate and acetate, could induce Tregs in the colon. This is in germ-free mice, and this was in a GPR 43-dependent manner. And then these two other papers that came out in Nature, probably concentrating more on butyrate and on the histone deacetylases for the induction of regulatory T cells. And we absolutely support this data through our studies. We looked at particularly high fiber type diets, acetylated resistant starch diets, and could get really beautiful levels of acetate in the blood and in the feces. And we used these, and similarly, we were able to see this remarkable induction of Tregs in the colon, using here a FOXP3 reporter mouse. You can see it was so obvious which mice had been fed acetate or which mice had been fed a very high fiber diet by their obvious Treg numbers in the colon. But a little bit different from some of those reported papers, at least with our diet, we could get really high levels of Tregs also in the periphery, at least in the spleen and some of the lymph nodes, as well as very high levels in the colon. And interestingly, we saw a reduction in Treg numbers in high-fat fed mice. So it could be that high-fat feeding, typical of Western diets, might be anti-tolerogenic and feed into some of our thoughts about what might be causing Western lifestyle inflammatory diseases. So when we looked at the microbiota in our very high fiber fed mice, which were producing really high levels of Treg, we got slightly different results than what's been published, or at least by some groups. We saw a fantastic expansion of Bacteroides in these high fiber fed mice. And we saw, in fact, also a reduction in Clostridial species, which have been the ones more associated with Treg numbers. And our results support at least one paper from Jeffrey Gordon's group, which have also associated Treg expansion with Bacteroides. But we wanted a practical outcome for inducing tolerance with being a pro-tolerogenic diets, and that was in type 1 diabetes. And similar to all the other studies I've mentioned, we get a really nice protection from diabetes in these very high fiber fed mice. And I'm not, I think, not shown here. We're showing that this is mostly, or in a large part, GPR43 dependent. This is incidence of mice progressing to diabetes in the NOD mouse. And you can see the nice protection typical of a mouse on high fiber versus the inflammation you can see in the islets on a normal NOD mouse on a normal diet. And what we could show using, this is somewhat complicated, but I'll try and explain it, but you can use a NOD 8.3 mouse, which has a T-cell receptor transgene, which is specific to an autoantigen, and you can transfer these cells into mice that have received these different diets, and these mice that have received these diets have expanded or not Treg numbers, and you can show that in the beautiful, this particular diet that is fantastic at inducing Treg numbers, that you don't get proliferation of this autoreactive transferred T-cell subset, showing that the Tregs are containing its proliferation, whereas you don't see this, you see this beautiful proliferation, particularly in the 43 knockout mice, because you don't have this anti-inflammatory receptor protection, also in control mice. So it's really illustrating that we do see peripheral immune tolerance or suppression in this system. So I'll finish there. I really strongly believe that many of the metabolites from bacteria and also from non-bacterial sources acting through these pathways I've mentioned, such as the metabolite-sensing receptors, also through transcription factors and through epigenetic or histone deacetylase inhibition, might be accounting for the beneficial effects of many of the metabolites. So in fact, there's a human experiment that sort of is hinting at to some of the things I've mentioned. And in fact, that there's these blue zones where people have extraordinarily longer lives, such as in Sardinia and Icaria and Okinawa. And there's something that's common to all these diets, and that is, I suppose, consumption of many of the healthy sort of foodstuffs and metabolites that I've mentioned. For instance, omega-3, which I haven't talked about at all, but is anti-inflammatory, binding through GPR120, tryptophan-rich foods. Even wine can produce acetate through its metabolism. All of these places eat lots of vegetables, nuts, and dietary fiber, and generally low consumption of all the things that probably disrupt homeostasis, such as high fat and high sugar and caloric restriction. So I'll finish there and just briefly mention the main people, the three main postdocs here, Laurence, Alison, and Eliana, that did most of the work I presented here. The work was started by Kendall when she was a PhD student in my lab some years ago. We've been supported with the diets through the CSIRO, various collaborators from around the world, including Sidonio, who's here today. Thank you.